And now it's my pleasure to introduce the last speaker of the session. And I don't need to read who she is, since we have known each other since we're 15 years old in high school. So Audrey is a Canada SciFar III chair, and she's also an assistant professor in the computer science and computer engineering and electrical engineering departments at Université Laval, and also the leader of the methods research axis at the Institut for Intelligence et Données at Université Laval. Audrey.
Thank you, Flavie. Hello, everyone. It's a pleasure for me to be here today. It's actually my first time in person at a conference since 2019. I hope you can see me behind the huge desk. When I see desks like this, I always wish they had included either a box or some, these huge chemistry and physics books from high school that I could step on and be the same size as the other speakers. So today I'm going to talk to you about a line of my work that I've been conducting with Flavie and our students. The goal in this work is to use reinforcement learning to support research, assist research in their research duties, in this case, to operate imaging devices. And the idea is, well, maybe possibly with these tools to assist them, maybe there would be potential to accelerate research in neuroscience by helping researchers to acquire good, even better images of structures that are very hard to capture and observe, even with the amazing devices that microscopists are using these days. Okay, yeah, so kind of more specifically, the talk today is going to be about STED microscopy optimization. So you might not be familiar with STED, so I'm going to give a very brief overview of the thing before we start talking about the methods. The STED microscopes essentially produce images by using pixel by pixel scanning of biological samples using two lasers. So if you want to produce a nice image like the one you see here, you'll have your biological sample, let's say a neuron, and you're going to throw in a bunch of fluorescent molecules in there. And the property of these fluorescent molecules is that they can be excited by some wavelengths and start emitting light. So we'll have this first laser that's going to be called the excitation lasers. So when you go pixel by pixel over your biological sample that has been populated with these fluorescent molecules, is going to turn on the molecules, and then you can capture the structure that you're looking at by looking at the light that's being emitted. Now, if you're looking at a very, very tiny structure, what you'd like is you'd like this laser beam to be with the smallest radius as possible, just to turn on the molecules in the very tiny region that you're trying to look at. Otherwise, you won't be able to see your structure at a good resolution. But the problem is that with the limit of diffraction, you cannot have that laser being tiny enough just to look perfectly, precisely at the structure. So you're going to use a second laser that's going to be shaped like a donut. And the second laser has also this nice property that instead of exciting molecules, it is turning them off. And since it's shaped like a donut, it's going to turn off the molecules that were like in the exterior radius of the initial excitation beam and leave only on the molecules that were in the center of the beam, which is going to act as if we had turned on the molecules initially with a tinier laser. Roughly, that's my explanation of what's going on here. I'm not the expert on that side, obviously, but with that neat trick, this allows us to reach high resolution imaging that's below the limit of diffraction and observe structures that are at the nanometric scale, which we refer to as nanostructures. And this is cool because with this technique, we're able to observe things, to distinguish structures that would not be distinguishable using classical microscopy techniques. which are limited by the diffraction. However, using these systems is not necessarily intuitive. As you can see, this is an example of the very non-intuitive graphical user interface that the microscopist has to use if they want to use the system. So you see there's a lot of parameters in there that needs to be tuned. And even if we were only to focus on few parameters, basically the ones that just control the two lasers and maybe imaging time per pixel, given that there are multiple objectives to take into account where you acquire an image, even if you're going to restrict the number of parameters that you're going to tune and just rely on default values for the other ones, acquiring a good image with this system requires a lot of expert knowledge. This is something that someone who did a maybe master's, PhD, postdoc, and has been working in the field for a long time can do, but with a lot of effort. It's something that would be nice if it could be done by graduate students at the lower level, let's say master's students or even undergrad interns. So this is a hard task, and it's also a task that needs to be done every time you want to imagine a new sample, because biology has some randomness, and it's very annoying. If you tune your system, it's not going to give you good images on all samples. It's not even going to give you images on all samples of the same kind. Like if you're looking at a particular type of neurons, the neuron culture from today will require different parameters to be imaged properly compared with the neuron culture from tomorrow, which means that tuning of these parameters is something that needs to be done on every biological sample that you want to target, which leads to a lot of waste in terms of samples that could be used to image and actually conduct research, because they're being used to just tune the parameters. And so so when you tune these parameters, you're looking at a lot of different objectives. Just getting a good image with high resolution is not enough, because sometimes the parameters that are going to give you the good image, they are also very, they can be very destructive on the fluorophores, and they can cause your fluorophores to just, let's say, die, and you won't be able to take more than one image at the same location, which is annoying if your goal is to track some sort of behavior that's happening over time. You'd like to be looking at some place and see how it changes. But if you can take only one image because this one image just destroyed your sample, well, you didn't win. So yeah, finding the good parameters is hard. Finding good parameters that do what you want to do within your constraints is even harder. So there would be a lot of potential benefits of automating maybe some parts of this parameter tuning to help the researchers acquire these images. So today I'm going to present to you some work that we did to develop strategies that can tune STED parameters automatically. Yeah, let's say automatically to some extent, as images are being acquired. So we can formulate this problem of parameter tuning as an iterative game where you have an automated agent which sequentially recommends parameters to use. So it can send parameters to the interface. These parameters, they can depend on different things. They can depend on a low resolution, cheap to acquire preview of the sample. It can depend to the previous parameters that have been tried on the biological sample. So the agent sends parameters to the microscopes. An image is being acquired, and then some feedback can be computed from that image, and that feedback can be sent to the agent. The agent uses the feedback to improve and can propose, again, the next parameters to be used for the next image, and so on and so forth. So the idea here is that the agent is going to adjust the parameters based on the previous trials that it's done, so that it's going to improve over time through what could be viewed as some sort of trial and error process. It tries something, sees some feedback, improves, The important thing here is that unlike supervised learning, the agent has never been told what would have been the best thing to do. It can only compare different things that it has tried in the past and see if it is improving, but it cannot know if it is doing the best. It can only know if it's doing better. And this is let's say the hard part of those kind of iterative learning processes. So yeah, the agent is basically just going to learn to image the same way that an expert in microscopy or a poor lab intern would try to do, which means it's going to try things, observe, and then improve. And so with these strategies, we aim to reduce the impact of the tuning in basically, I would say, two aspects. On one hand, we hope that it's going to reduce the images that are wasted because they were used for tuning, but they resulted into bad images that cannot be analyzed and used for research. And also, ideally, we want to find for imaging parameters that will have low impact on the fluorophores, on the fluorescent molecules, which would allow to acquire more images at the same location and monitor changes over time. So this game here, well, this can be seen as a reinforcement learning problem, where in the reinforcement learning jargon, we say that our agent selects actions. So the actions here are the imaging parameters given some state. State is basically the sample and the history of the parameters that have been tried. And the feedback can be seen as a reward signal, so signal that the agent is trying to maximize. This is what the agent integrates for learning, and possibly the next state that would be generated by the following the imaging. So if we were considering, let's say, imaging always at the same location to monitor something that can change over time, the next state could be how the things look after the agent has taken the image, which could, for example, contain the fluorescent molecules that have been destroyed by the imaging process. So this way the agent can see its impact on the system itself. So this kind of reinforcement learning problem, it's nice because you can tackle problems that are sequential. So you can have your action at time t that is going to influence your sample, make you transition in a new state, and then eventually you can stack up those rewards. But the problem is that optimizing a system like this one may take a lot of samples. So in the first step in the project, instead of considering this formulation, we rather focused on a simpler setting, a simplification of the RL setting that's called Bandits. Funny name, I know. And it's this, in this simplification, well, it's like there is you can remove this notion of state. So you don't care anymore about the future impact of your action. Let's say we just focus on the immediate reward value that was generated. This is a fine enough formulation if we assume as a starting point problem that each image is going to be acquired at a different location in the same sample. So we're going still to try to minimize that photobleaching effect, but not with the intent, actually in this example, not with the intent to take multiple images at the same location, but just for the problem setting to fit. We're going to suppose we're going to image at different locations at every time step t, which means our agent can now focus on just the reward, not care really about the transition. Yeah, and even in this simple setting, there are still challenges. Most importantly, the challenge that we have multiple objectives to optimize, which is why we decided to simplify the problem in the first place, because this challenge in itself was not easy to solve. So as I said earlier, there are two things that we want to do. We want to acquire good images, but we want to do that in a way that is the less destructive as possible for the fluorescent molecules. So we want to maximize the image quality, being able to acquire good images, images with good resolutions that allow us to distinguish the nice structures. And at the same time, we want to minimize the impact that we're having on the fluorescent molecules. So making sure that after we do an image, there remains as many molecules as possible, so that we would be, in theory, able to take more images at the same place. So the first loop that we implemented looked like that. So we had our agent whose goal was to recommend parameters for imaging. And those parameters were used to produce an image. And this image was used in turn to produce two reward signals. One reward signal that was associated with image quality. So in this first loop, we had an expert in the loop looking at each image that was being produced and giving it a score. Again, the goal here is to have a system that assists the experts in doing the work, not to replace them. So in this first version, it felt totally fine to have the microscopist with the algorithm doing the work. So we had that person used to be Flavie, looking at the image, giving a score. We had the image being sent to an online analysis tools to compute the amount of photo bleaching, and these two reward signals were sent back to the agent. Now, there is this question of how to articulate the preference between the two objectives. We want to maximize one, we want to minimize the other. It would be simple if one could be maximized while the other is minimized, but in practice, that is not the case. It's a trade-off that you want to make between these two, and it's very unclear how this trade-off should be done. It depends a lot on the task at hand. So at first, we decided to bring in the expert in the upper loop, so in the parameter selection process, to do the articulation of the preference. Anyway, the expert had to be at the microscope because they were rating the images, so might as well use them at the parameter suggestion step 2. So that means that at this stage, the agent was not completely autonomously proposing the parameters to be used. It was rather proposing to an expert a bunch of options, like compromise options, that it could guess could happen or could be achievable with the system. And by looking at these different trade-offs that the agent was telling the expert, look, according to what I've seen until now, I think these are the trade-offs that are achievable with the machine on that samples. You can either get this quality versus this photo bleaching, or that other quality against this photo bleaching. What do you prefer? And the expert would look at these trade-offs and pick the one that they found the most acceptable given the situation. So now we had the expert doing a lot of work here in the system. And exactly how did the agent propose these options? For that, we relied on very classical Gaussian process kernelized Bandit algorithms. It's just that usually when you have a single objective, the agent would model, it would learn the model for the reward function for your single objective. And at the time of decision, it would, let's say, sample from that function and then just do a max to pick the action, or in this case, the parameters that maximize that function. In this case, since we have a bunch of objectives to model, the agent would build different models, and it would build the different outcome options from these models and present these outcome options to the expert. So we really integrated the expert in the loop of, let's say, parallelized Bandit algorithms that would learn parallel models for the different reward functions considered here, one for each objective. But that was a lot of work for the expert. I mean, yes, the goal is to develop tools to assist experts, but at that point, it didn't feel for the expert like they were saving a lot of time and being helped that much, besides maybe that the system was able to converge to interesting parameters that they wouldn't have converged to otherwise. So in the next step, after running with this scheme for a while, which allowed us to collect a lot of data, we were able to use that data to train neural networks to replace the expert at the two different locations in the dynamics where they appeared. So for estimating the image quality score, we trained a very simple, small convolutional neural network that could take as input an image and output the quality as the expert would have rated it. So the expert didn't need anymore to be looking at images and provide a quality score. That could be done automatically by a CNN. And for articulating the preference between the different outcome actions, we trained a small neural network, a fully connected neural network that can take any pairs of two outcomes and produce an ordering of these different outcomes trade-offs, and be able to articulate the preference the same way that it has seen the expert do in a lot of the experiments where the expert was doing that thing manually. So that gave us what seemed to be a fully automated system, which was a nice first step, and we tried it in practice. So on a real STED microscopes, we tried optimizing 3 parameters, which if we discretize them, led to 1,000 configurations, possible configurations of parameters, where we consider the power of the 2 lasers and the time spent per pixel during imaging. It's only 3 parameters, and they're discretized here, but even if you do that, it's still a lot of combinations, many more combinations that you would be able to try if you were to tune that by yourself on a single biological sample. And the experiment went as follows. So for each biological sample that we considered, each step meant taking first a confocal image, which is classical microscopy image that is low resolution, but very cheap to acquire and non-destructive. Then we would take a first STED image using the parameters recommended by the agent, and then a second STED image using the same parameters, and we'll be taking 2 STEDs because this allows us to measure the amount of photo bleaching that occurs between the 2 images. And so the goal was to maximize the quality in the first STED and to minimize the amount of photo bleaching, which we computed using the 2 STEDs. So here's a snapshot of the results that we got with this. So each column is for a different experiment on the different biological samples. The first row shows images at the beginning of the optimization process. The second row shows images at the end of the optimization process. And the lower left corner always shows a part taken with the confocal image. So by comparing the lower left corner with the rest of the image, you can see the gain in resolution. And on each image, you have the quality that was given by the expert on each of these images. So you can look at you can first look at the quality scores and see that the quality scores on the bottom row are always higher. So the quality was able to improve over the optimization process, in all cases. If you look at the first column, you can see that by the end of the optimization, resolution and parameters were good enough such that we were able to see structures. So in this case, for the first column, the actin, it's structures that when you observe it correctly, you're able to see that it has some shape in forms of shaped like a ring, and we can see these rings well on the bottom image towards the end of the optimization. So the system was able to converge towards a region of parameters that allowed to observe that. And if we look at the 2 other columns, we can also see the improvement in resolution between the beginning and the end of the learning process. So in the second column, you can see that the fibers are much thinner towards the end of the optimization, so we can see them better when we compare with the confocal part. And in the last column, we can see that the tiny beds appear as closer to the shape of what would be expected with tiny beds. Now, in this Bandit formulation, the agent considers each biological sample, that is each image sequence, independently, like it's a different problem. Every time we want to tackle a new biological sample, we restart the agent on that sample, and that's a new game. And the task to identify optimal parameters for this given sequence is considered a different game compared with the next biological sample, meaning that each new optimization process will start from scratch with its own specific goal for that sequence. And knowledge will not be reused from one sequence to another. So you cannot tell your agent, you imaged something that looked like that yesterday. Maybe you can pick up faster what's going on if you see that things are behaving the same. Like, you cannot give it knowledge like that. Each new biological sample is a new game that is being played from scratch. Given the good results that we saw in the Bandit formulation, that motivated us to continue with the true sequential reinforcement learning formulation, where the task would be to learn a policy that can conduct efficient parameter tuning on any new sample by leveraging information that would be previously acquired or encountered in the sequences of image that had been seen previously on different samples. So share information about about more sequences on biological samples of the same nature, but maybe with slightly different behaviors across days, or even share information across biological samples of different nature. So now in this formulation, rather than seeing each biological sample as an independent problem, as a new game, we can see each biological sample as a new episode of the same game. So it's like saying we're trying to master the game of chess. Each biological sample is playing a game of chess against a different adversary. Eventually, if you play enough games, you're going to be able to draw some similarities between some of these adversaries and reuse your knowledge. So this is basically the approach here. However, if you want to solve these kind of problems, like I was telling you earlier in the talk, it takes a lot of samples, or at least just to design the methods and make things working and just see if there's a potential for things to work with this. It can be very difficult. So to do that, and do that in a non-destructive way that wouldn't cost us thousands of dollars of failed experiments, we started by designing a simulator. It's in Python, it's available. I'll give you the link at the end. A simulator of a STED microscope. It's called PySTED, hence the Python. And it can simulate both the material aspects of a STED microscope, like the detector, the lens, and the lasers, and biological aspects, which means you can give properties to the fluorescent molecules. So you feed that PySTED simulator with a data map that describes the positioning of fluorescent molecules in a virtual sample for which we want to simulate the imaging. And using that data map, you can turn on the excitation laser to simulate a confocal image like classical microscopy, low resolution. And you can, on top of that, use the simulated depletion laser to produce a simulated STED image. So we see here that the STED image has better resolution compared with the confocal image, as expected. And it can even simulate the photo bleaching. So here you can see the amount of molecules that would remain after the confocal image has been taken and after the STED image has been taken. And you see that after the STED image, there's not a lot of molecules remaining for this specific simulation. So we can simulate the photo bleaching phenomenon also. And the versatility of the data maps, well, they allow us to basically simulate any kind of structures that we want. We just have to put in numbers in a matrix and say that you have these molecules here, and they're going to be simulated. So one thing that we decided to simulate is nanostructures on a dendritic spine. So you can see here like the real image, kind of real image that you would get with a real microscopes, and what we got with our handcrafted data maps for that simulation. So it's in this case, it's not like we did put a lot of effort into making a realistic data map. It was very quick to do, and we managed to get something we decided was close enough for the first developments and the first trials that we wanted to make. And the nice thing, if you're going to work with a simulation like this, if you are going to design the data map by yourself, it means you know, because it's a simulation, you know where the true positions of the nanostructures are. So what you can do is that you can, given a stead, you can compute a score, like the F1 score, between the positions that you detected on the stead for the nanostructures and the true positions where they were located in the data map. And you can know how good is your stead for identifying the locations of these nanostructures, which, oh, maybe I should go back, which correspond to the very bright spots here. So and you can even do that in a very automated way. You can have, you can just detect the locations on the stead based on the local high intensity maxima, fit some Gaussian models on that. And then based on how far apart the Gaussians are, you can identify these peaks as being nanostructures or not. And then you can use classical algorithms to match the nanostructures that you detected to the true nanostructures and decide if whether these are like either true positive detections or false positive or false negative, and based on that, compute your F1 score. So all of that, you don't need anyone, you don't need a human to do that. It's an automated box. So that's nice. It can quantify directly the quality of your stead. So with that in hand, we decided to use PyStead to learn a sequential parameter tuning strategy under the reinforcement learning setting, where here the agent would have to learn how to adapt the imaging parameters over sequences of images, such as to acquire stead images where nanostructures can be detected and located. So in this setting, the value of an action resides not only in its immediate reward signal, but also in every reward signal that becomes available afterwards, thanks to the action that we took now. So this translates into the agent aiming to optimize at time t, what we call the sum of discounted rewards. So it's the reward signal that they get now, plus a fraction of the reward signal that they're going to get in the next step, plus a smaller fraction of the reward that they're going to get afterwards, and so on. And so this means that the agent is not learning now a myopic decision policy, but really a sequential policy. It considers the future when making its decision for the current action. So the complete reinforcement learning goes as follows. So each episode is like a different biological sample that we consider. We assume that here in this formulation that we would be taking 10 images per episode per biological sample. And we define the state as being the current confocal, so the low resolution, cheap acquisition, non-destructive microscopy technique that gives you an idea of what the situation looks like now, plus the previous pair of confocal stead that was obtained. So what happened at the step right before, plus some, like, let's say a matrix of the history of the past in this episode. So what were which parameters did we try previously in the current sequence? And what were the objectives that were computed for these parameters? So the objectives here, what we considered were the signal to noise ratio, the resolution, and the photo bleaching. So basically that matrix is like the memory of the agent in the current sequence, and the memory of the agent over the sequences is going to be stored in the agent's model that we're going to see right afterwards. So the action here are the imaging parameters. Again, 3 parameters that we consider: the power of the 2 lasers and the time spent per pixel. And in this first formulation, we considered directly the F1 score as the reward given to the agent to characterize the goodness of the stead image that's being acquired with these parameters. So here's the model that we used. So we used the PPO reinforcement learning agent, which is a state-of-the-art policy gradient reinforcement learning agent. I'm not going to go into the details of this, but it learns a model. In this case, the model is a deep neural network model that learns to map the state to a distribution of probability over the action space. And you can sample, for example, from this probability distribution to get the next action that should be played. So in this case, our neural network had 2 inputs. It had one part of the input to handle the visual information of the state. So a small CNN that took as input the current confocal and the previous confocal stead pair, and also a fully connected neural network that was handling this matrix of the history in the current episode. All of this was being processed, the representations were being concatenated, and eventually at the output we're outputting the distribution over the parameters to recommend. In this case, I just presented it as it recommends directly the parameters to be used. So it can either output a distribution or can output a suggestion by itself. But yeah, the idea is that it takes the state as input and it outputs what you should be doing. And to learn that model, you use the rewards that are being sent to your system. So in this first formulation, the rewards were directly the F1 score. So it recommends some parameters, you take an image, you take that stead image, you compute the F1 score, and then you send back to the model, which basically tells the model, good job, you picked good parameters, or we're not able to see any nanostructures here, try again. And don't try that specifically again. So if we look at the first results that we got, so episodes of 10 images each, and we did 20,000 episodes, so that gave us 200,000 images total. This first curve shows the rewards that were being collected over the different episodes. So you see that the reward curve is going up, which means that as the episodes were progressing, we were getting better and better rewards, which means better and better F1 score at detecting the nanostructures locations. And also, it's normal not to get a perfect F1 score. Perfect F1 score over 10 images would be 10, because the maximum value of F1 score is 1, and you're computing it on 10 images. The perfect value, if an episode was to give the perfect F1 score on all images, would be 10. It's normal that we cannot reach 10 in a single episode, because you need to think that the agent always has to at least make a few tries in the beginning to just figure out what kind of samples they're tackling in their game, what kind of adversary they're playing against. But the fact that we were able to reach almost like 8, which means that it probably was using maybe 1 to 3 images on average to figure out the kind of samples they're playing against. And after that, they were able to adapt pretty quickly. If you look directly at the value in terms of F1 score, you averaged over episodes, you see that this aligns with what I was saying, and that meaning that towards the last episodes, the agent was able to get very few variants in the F1 score of a single episode. So probably spending the few images of the episodes figuring out how this biological sample works. And as soon as it was able to understand what's going on, it was able to quickly shift through imaging with good parameters. So overall, F1 scores of acquired image get better as the agent learns, which is what is expected. And now if we look at how this translates into objective values, so we want the value for the resolution to be as small as possible, because the smaller this value, it means the more we are able to distinguish structures that are close to one another. So we can see that resolution improves over time. The signal to noise ratio, we want that to go up. So we see that there's a slight improvement over time. And more importantly, all of this happens while the photobleaching remains stable. So this is these results are very encouraging. But if you remember, the reward signal that we're sending to our agent is based on the F1 score, which means that we need to have access to the true location to actually send feedback to the agent, which is annoying because in practice, we want to know where the nanostructures are. We don't know where they are. If we were able to compute the F1 score, we wouldn't be imaging that because we would already have the answer. So it's nice for a simulation and a first proof of concept, but you cannot implement that in practice. So in the next step, we thought, why not look at some reward functions that could be implemented in practice and see if we can get a good behavior also with these other reward functions that could be usable. So what we tried is that we made an expert image data bank. So a bunch of images that were taken by an expert and that were ranked in order of like which one's the best image and to which one's the worst image. And we trained the system to be able to rank any new acquired image compared with this previously acquired bank. So that is something that we can do in practice. Once we've made the bank and we've ordered a bunch of images, we can have any new images come in and have that system say, okay, I think this is like the third best images of your bank. Okay, perfect. That gives you a reward. It's not the best. If the image was considered the best, you give it a reward of 1. If it would be considered the worst, you'd give it a reward of 0. If it's like the third best, it's a reward of 0.7. Easy. And that can be computed because that does not rely on the true positions of the structure you're trying to image. So the higher the ranking of the acquired STED image, the better the rewards. So that kind of makes sense also with the intent of the user with that system. So now if we look at the experiments conducted using that reward function, so we cannot compare the curve on the rewards with the previous one, because although it goes on the scale from 0 to 10, they're not the same nature of rewards, but we can look at the shape of the curve and it resembles what we had previously, which corresponds to basically the agent collecting more and more rewards over episodes as the episodes proceed, which means the agent is getting better at its task. And if we look at the other indicators, so because we're in a simulation, we can compute the F1 score just to analyze here. We realize that this this reward function based on the ranking of the image compared with other previously acquired images allows to reach a good F1 score also over time. And in terms of the other objectives, we see that it allows to improve the resolution, it allows to improve the signal to noise ratio, and it can keep the photo bleaching stable. So we were really happy with that. If you look at the actual image sequences that it can produce, you can really see that over time, it is able to find parameters that can really highlight these nanostructures. So it's a simulation. It's still a simplistic simulation, but it's a good first step in that direction. So right now we're in the process of seeing how can we generate simulated maps that are going to lead to images that are going to be more realistic, closer to reality. At the same time, how can we translate this to a real STED microscopes? Can we translate directly policies that have been learned here and just fine tune them? Or will we have to train policies and learn policies in simulation that are more robust and that cannot perform as well, but still can be transferable? So a bunch of new challenges related to transfer from simulation to reality awaiting in front of us. So I'm going to end on that, thanking all the collaborators, the student working on the project. And as I said earlier, the PySTED environment is available online, and we have it wrapped in a gym environment. So if you are looking into doing reinforcement learning and you'd be interested in trying out our environment, well, feel free, it's there. You can, I know the address is probably not easy to grasp on the slides, but if you just Google gym and PySTED, it should be able to find. Thank you.
Thanks a lot, Audrey.
Thank you for the great talk. I think it has great applications. So I was wondering how the agent learning speed compared to human. For example, a pre-trained agent compared to a fresh graduate student, or a well-trained agent compared to a postdoc, for example.
That's a good question. How do they compare with humans? So if we consider the sequential policy that was the one that I was showing in the last examples, it seemed like that agent is able to learn good parameters within very few images. So that seems to be really fast. But I don't know how experts would behave in that simulators, because when we look at these results, we think, cool, the agent is able to find good parameters using 2 or 3 images. That seems to be very quick. But we compare that with numbers that we have for humans, tuning parameters in the real stead, where the image is probably more noisy than what we have here. So it's looking good, but I wouldn't say at that point that it's going to be better than an expert. Better than a graduate student, probably. Definitely better than a summer intern, even on the simulator.
Great, thank you.
Yeah, we have a question from the chat from Roberto Araya. He thinks these are beautiful images and suspects that these images might be taken from neuronal cell cultures. If that's the case, could you elaborate on the challenges in this approach, like this RL approach, if you use vivo 2P STED imaging?
Okay, I'm not the good person to answer that question. That's a technical question on the neuroscience side of the project. I don't know if the question refers to the difficulty of producing the samples that would help the agent learn, or the difficulty to image on these samples. What I can say is that the agent is not like it's not like in active learning where the agent asks for a specific data point. In this case, the agent takes the sample that are being sent to him. So the agent is never going to ask you to produce a given sample for them. If that is what the question was about. But yeah, if the question referred to a sample that is particularly difficult to image or particularly different from the previous samples, if it's because the sample they're talking about is very different from the other samples that have been seen, it's going to depend on how, like, what was the variety of samples that had been seen in the training of the agents. For now, well, this sequential agent, we've trained it on the simulation of the nanostructures on the simulated, very simplistic simulation of a dendritic spine. So I would not be able to say confidently that these would generalize to the kind of structures they're talking about. But yeah, eventually, as we build more, as we build simulations closer to reality, we can start trying with samples that are more difficult. So if Flavie has grasped that question, we can look into the difficulty of that, those kind of samples, and see if we could integrate these in our experiments at some point. Thanks for the question.
Thanks for a great talk. I was just curious if you've tried to implement a penalty on some of the parameters that an expert might have identified as being likely to cause photo bleaching, if that's something that you've explored.
So yeah, good question. That could help make the learning faster, but the more we bring in those kind of tweaks, the more we constrain our agents to do something the same way that the humans are doing it. So the way we tried right now is that we gave complete freedom to the agent within the constraints of the parameters. Like we set the constraints of the parameter spaces to something that would not allow the agent to break the system. For example, in the first application where we deployed it on a real estate microscopes. But besides that, we really wanted to give that complete freedom to the algorithm with the hope that maybe it would be able to discover a way of doing things that's very different from the way we do it. So yeah, applying penalties to actions that make less sense from an expert perspective might accelerate learning, but it could prevent us from learning alternative imaging strategies that would be like very, very new.
Thank you for an amazing talk. I had a maybe a naive question. I was wondering, to increase the realism of these images, did you also consider maybe adding a component using adversarial learning, some sort of like GANs or something, to try and see whether that can help you as an additional step where you'd...
So adding GANs, it's not clear to me where we could add GANs in the reinforcement learning dynamics, but we are using GANs to try to produce images that look more realistic. So GANs are going to be part of the playground tools, but not in this learning dynamics. This learning dynamics is really through feedback, because the agent is not trying to mimic something that we know.
Yes.
And the goal of the GAN would be to try to force it to mimic something that we know. And yeah, this goes against what we're trying to have it do. Yeah, we wanted to get good results. So we wanted to come to the same finality as we would with a human expert, but ideally using a different pathway to achieve that, so that hopefully the pathway of the agent could have some benefits in terms of the objectives.
Great. Thanks again, Audrey, for your talk. And now we will have a very short break and come back for the panel discussion just after.